My name is Professor Wigneshweraraj. I'm a professor of microbiology at the MRC Center for Molecular Bacteriology and Infection at Imperial College in London. We work on bacteria. In particular, we are interested in how bacteria switch on and switch off their genes. For switching on their genes, bacteria use and depend upon an enzyme called the RNA polymerase. So what we are interested in is how the behavior and the performance of the RNA polymerase is controlled by all the various factors that the bacterial cell perceives as it grows. Now, because the RNA polymerase is an essential bacterial enzyme, there are several thousand copies of this enzyme in the bacterial cell. Now, what the Nanoimager allows us to do is to study this enzyme at a single molecule level. So what we can do is that we can capture and visualize how individual molecules of this RNA polymerase behave as the bacteria perceive different signals in its growth environment. This single molecule microscopy is quite revolutionary. It's in fact a game changer for us, because when I was young, the microscopy occupied an entire room of particularly this size. Now we have this capability in a single small box, and it's simple to use, and the software is relatively easy to use as well. A major advantage of working with a company like ONI is the access to the founding scientists of this technology. This allows us to discuss results with them and perhaps help them to develop the technology which is of equal benefit both to them as well as to us users. And this is an exciting opportunity to build this technology further.
The Nanoimager is great, especially if you want to look at the function of an unknown protein. We can tag that protein with a fluorophore and then look at it at a single cell level, how it diffuses, its activity, its expression levels. We all know that cells aren't uniform. They can be expressed and diffuse at different levels between different cells.